This condition of photorespiration just happening was the default for a very, very long time, until flowering plants came along and devised a couple of different strategies for coping with photorespiration. There are three ways to deal as a plant. You can be a C3 plant, or a C4 plant, or a CAM plant. C3 plants don't really do anything to deal with photorespiration. Under very hot and dry conditions, plants just close their stomata to prevent dehydration. Oxygen builds up, carbon dioxide levels drop, photorespiration happens, and that's it. For an individual plant in a particular site, they, in essence, do nothing to deal with photorespiration. But over the long run, this affects where different species of plants are capable of establishing populations that can persist. C3 is the default condition. Most plants just suffer through it. But there are other ways. C4 plants have a specialized structure in their leaves called krantz anatomy. Krantz anatomy means there are specialized tandem cells, normal mesophyll cells, and bundle sheath cells. These cells have an additional enzyme called PEP carboxylase. This enzyme is, like rubisco, a carboxylase, but unlike rubisco, it's not an oxygenase. PEP carboxylase can fix carbon dioxide even at low concentrations. In the mesophyll, carbon dioxide is fixed to a 3-carbon molecule, temporarily making a 4-carbon molecule, and that's how we get C4. The 4-carbon molecule is transported into the bundle sheath cell where the carbon dioxide is released. This way, carbon dioxide concentration is kept high in the bundle sheath cells and photorespiration is reduced. This costs some ATP, but it's not as wasteful as photorespiration when it's very, very hot and very, very dry. Thus, oxygen is separated from rubisco spatially. In this slide, the krantz anatomy is shown. The top cell on the right here is the mesophyll cell with PEP carboxylase, which scavenges whatever carbon dioxide may be around and fixes it into a 4-carbon molecule with a familiar name, oxaloacetate, which we saw in that other cycle. The same with malate, which we also saw in that other cycle, the citric acid one, which is very different from the Calvin cycle. Malate is then transferred to the bundle sheath cell, so named because these cells sheathe the vascular bundles that move the sugar to other parts of the plant. In the bundle sheath cells, the carbon dioxide is released along with our old friend pyruvate. Pyruvate can then return to the mesophyll cells where it is phosphorylated to form PEP once again. PEP stands for phosphoenolpyruvate. These C4 plants can grow better and faster in places where the conditions are right. Many of our important crops like corn and sugarcane use this strategy. Another plant with C4 metabolism is crabgrass. This is why, in the springtime, our lawns may look beautiful and perfect, but as spring turns to summer, the crabgrass starts growing much more quickly and better than the other lawn grasses, which are C3 grasses. A third way to avoid photorespiration is called crassulation acid metabolism, or CAM. Note the background image. CAM plants open their stomata at night and close them during the daytime. At night, they take in carbon dioxide and fix it as organic acids, for example, crassulation acid, which is unstable, but it can release the carbon dioxide during the day when there is sunlight to provide the ATP and NADPH. In a way, CAM plants take a long, deep inhale of carbon dioxide at night and use the gas during the daytime when they have the power to do something with it. So unlike C4 plants that have a spatial separation of oxygen and rubisco, CAM plants have a temporal or time-based separation. CAM plants include many desert species, like cactus, though pineapples and many orchids also have this ability. This slide compares how C4 plants and CAM plants grow well in conditions where C3 plants don't grow well. In C4, it is a spatial separation of rubisco and oxygen in different cell types. In CAM plants, it is a temporal separation, day and night. As we close this chapter, it is useful to review why we spend so much time talking about this process that we as animals are ignorant of, at least at the cellular level. We depend utterly on photosynthesis to provide for us as interpreters of the sun's tremendous energy. Everything we eat either photosynthesizes or ate something that photosynthesized at some point. Every breath that we draw includes oxygen that was water split by the sun's energy in a photosynthetic organism. This process is very ancient and has literally changed the face of the planet. The advent of oxygenic photosynthesis turned iron-bearing rocks red and poisoned many oxygen-intolerant microbes and led to the production of a layer of ozone in the upper atmosphere, which shielded the land from ultraviolet radiation. The land was lifeless before there was an ozone layer. Our economy is dependent on the energy stored by ancient photosynthesizers. Here in Alabama, we have great coal deposits in the middle of the state and petroleum reserves in Mobile Bay. Both of these are non-renewable resources accrued over millions of years through photosynthesis. And today, in addition to our food, plants provide us with cotton to wear, paper to write on, wood to build with, and much, much more. While we face a climate crisis caused in part by the increased concentration of carbon dioxide in our atmosphere, photosynthesis is the only natural process that can remove carbon dioxide, and it does it an awful lot. Here again are our learning objectives for Chapter 10, Part 1, and 2, and 3. And we have arrived at the end of Chapter 10 and Unit 2. So the next item on the agenda will be Exam 2, which will cover Chapters 6 through 10, which means the tour of the cell, membranes, metabolism, cellular respiration, and photosynthesis.